Welcome friends. You are probably wondering why I told you not to watch this video. I just wanted to do an experiment to see if people would still watch this video given the information of not to do so. And it's very funny if you look at the nature of human beings. We always want to try to do things that we are told not to do so. So I am asking for maybe for just one reason, if you can just comment in the comment section below why you chose to do so, given the warning of not to do so. So before us, we have this biology paper 2, and this paper is coming from the year of 2003, and we are going to answer questions from section A. We have question 1. Figure 1.1 shows a plant cell, and we know that whenever we look at a cell, we are looking at definitely a life. So the life of a living organism, whether a plant, animal, whatever, as long as it is under a living organism, it all depends on a cell. We have been given some parts here. We have a cell, we have structure A, chloroplast, and the structure B. Now question 1, which is A, identify and state the functions of the structures labeled A and B. So structure A, this is just a vacuole. Then we have to give two functions of the vacuole. The first one, we can say to store salt solution. Then B, we can also say to store enzymes. Then there are so many functions associated with this part. Then if you look at structure B is a nucleus. Then two functions of the nucleus. The first one, to control all the metabolic reactions that occurs in the cell. Then the second one, to store genetic makeup of a cell. Then we move on to question B. Under question B, list two differences between the plant cell shown in figure 1 and an animal cell. So the first one, we can look at in terms of the shape. We can now say plant cell is regular in shape, while animal cell is irregular in shape. The second one, we can say plant cell has a cell wall, whereas an animal cell does not. Okay, if you want, we can also look at the difference in terms of the presence of the chloroplast. We know that a plant cell has a chloroplast while an animal cell does not. Even the presence of cellulose in between the cell membrane and the cell. Then the function of the cellulose, it helps in the formation of cell. We have question 2. Figure 2 shows stages in the process of making yogurt using fresh milk. So yogurt is a substance that is commonly known. We know this. Now here we have been given some stages that are summarized, and these stages are there to give us the picture of what happens during the formation of yogurt. So yogurt is a dairy product. It means it is made from milk. Then we want to look at now what stages are involved whenever yogurt is made from milk. So the stage 1, we are told to heat milk slowly to boiling point. The second one, after this milk is heated, then we are supposed to allow to cool to lukewarm. Lukewarm is a favorable temperature. So it's a favorable temperature. So this temperature is neither high nor low. Then stage 3 is where the bacterial starter is added. And we need to look at what is the importance of bacterial starter. Then stage 4, pour the mixture into containers which have been thoroughly cleaned and rinsed with boiling water. Stage 5, after this is done now, you incubate the mixture of milk and starter for about 8 hours at 40 degrees. So this one is just you keep it for 8 hours. Then in stage 6 now, once the yogurt has set, place it in a refrigerator. So we have now, using the stages shown in figure 2 and your own knowledge on the topic, answer the following questions. Suggest any 2 ways in which other bacteria other than that in the starter in stage 3 is prevented from growing in the milk being used in making yogurt. So here we are looking at the formation of yogurt. Now the formation of yogurt is all about the process of anaerobic. So it's all about anaerobic respiration. Now this anaerobic respiration happens in presence of bacteria. Now we may have negative information about bacteria, but what I can share with you guys is that there are some bacteria that are useful, like the one that I used during the formation of yogurt. If you look at the formation of alcohol, also the formation of bread during baking. So to summarize, we are saying some bacteria are useful, then others are harmful, like the one that causes diseases. Now, if you look at the first stage, we told we need to boil, we need to heat the milk to its boiling point. So why are we doing this? So the reason why we are doing this, we want to kill the unuseful bacteria that may be present in milk. So that is why we did that. So 2 ways in which we can prevent those bacteria from growing is by we increase temperature. So increase temperature, because a very high temperature will inhibit the bacteria from growing. Then the opposite also is true, where we decrease the temperature. So for enzymes to grow or to respire anaerobically, we need to have a favorable temperature. So that's a point that we are trying to look at here. So this it's similar with enzymes. We know that high temperature denatures the enzymes, while low temperature will make the enzymes to be inactive. But for this one, it's all about growth. If the temperature is too high, the enzymes, the bacteria will not grow, and so is the over decrease of temperature. Then B, why is the milk cooled before the adding the bacterial starter? So here, because we know that the reason why we are cooling this milk before we add the bacterial starter is that we want to bring that high temperature to a normal temperature where the bacteria can do their work. So we can say to provide a favorable temperature. So to provide a favorable temperature for bacterial starter, we can just hear the word starter so that they can start the process of anaerobic respiration. So the whole thing about preparation of yogurt, is just from the process of respiration, which is anaerobic, and this one will involve the fermentation. So it's a fermentation indirect kind of process where the milk will be fermented, because in milk there is a sugar which is a lactose. Then that lactose, as long as there is the presence of bacteria, that lactose will be converted into a lactic acid. And if you look at the taste of yogurt, it has a sour taste, and we know that the sour taste can just be brought by the presence of an acid. So in yogurt there is lactic acid, which was brought by the process of fermentation from the sugar, which is the one we call lactose. So this process of anaerobic respiration and the presence of bacteria is just to convert that into the lactic acid. So what happens to the bacteria? So we say the bacteria will start to respire. Anaerobically, bringing the process of fermentation. Okay, then for D, what happens to the milk? This time is to the milk after the bacterial starter has been added and the mixture left to incubate in stage 5. So the milk will be fermented to produce lactic acid. This acid will lower the pH. Will lower the pH to cause the proteins to coagulate. So even here we have this coagulation process where we want now the pieces of the milk to stick together to form that solid substance we have in yogurt. So that is what we can say about that. But the point is, there is a lot that we can say about the formation of yogurt. So the best way you can do is you need to research, you understand more. Then question E, why is it necessary to rinse containers in boiling water in stage 4? So to rinse containers in boiling water in stage 4. So we can say to kill the harmful bacteria and prevent them from growing. Okay, so we move on to question 3. Question 3, figure 3.11 and 3.1.2 shows the experimental setup to demonstrate a biological process at the start of the experiment. So we have liquid J, liquid L, okay, then liquid M, then liquid K here. Initial, so this is the initial at the start, this is the level. Then partially permeable membrane, Okay, so now let's move on. Figure 3.2.1 and figure 3.2.2 shows the same experimental setup to demonstrate a biological process after 30 minutes. So let's look at what happens after 30 minutes. So we can see that at first the initial was somewhere here, even here. So there was a rise in the level of this liquid M. Now for this liquid to rise, there should be the movement of water molecules from one position to another position. So there is this process of osmosis. So we are dealing with osmosis. Now the difference between these two figures is that this one has risen more than the other one, because we can see, because this one is on this level somewhere here, and there should be the reason why there is a rise, a great rise than this one. Now let's look at the questions. So the questions is, two of the liquids used in the experiment were water. Okay, two were water and the other two were sucrose solutions. So we have a solution and water. From the experimental results shown in figure 3.2.1 and figure 3.2.2, name liquids J and L. So J, this is water. L is also water. Because the reason why we are saying this is water, this is water. This is a solution, and because of the solution, water molecules will move where there is too much water into where there is less. So water molecules moved in this direction. Even for this one, moved in this direction. So these two liquids were water. Explain why liquid M has risen to a greater height than liquid K. So why this one has risen to a greater height compared to the other one? So there should just be the difference in terms of concentration. So the one with the greater height has had a higher concentration, and because of the higher concentration, the rate of osmosis was higher than the one with the lower. As a result, it absorbed a lot of water molecules as a result of the increase in the height. So we can say that liquid M, so liquid M had a greater concentration, which resulted in a greater movement of water molecules. So that is the reason why. So we can say the liquid M absorbed more water molecules than liquid K. Okay, so we move on to B1. So name the biological process being demonstrated in the experimental setup. So this is osmosis. Then Roman number 2, how does the process you have named affect an animal cell when placed in water? So whenever we get an animal cell and place it in water, it is going to absorb a lot of water. So the animal cell will absorb a lot of water molecules causing it to increase in size, and eventually burst. So through the process of cell crenation. So that is what we are expecting to have here. Then we move on to question 4. Some married couples fail to have children due to females' failure to produce ova. In such cases, female fertility drugs containing a hormone are taken. Name a hormone that stimulates production of ova. So we can just look at this as follicle stimulating hormone. So FSH. Then it suggests why this treatment can result in the birth of many babies at once. So because this hormone can cause a female to produce mature ova, to produce 2 or more mature ova at once. So that's the reason we may have 2 or more mature eggs at once. Then B, females possess an endocrine gland that secretes the hormone you have named in A1 above. Name the endocrine gland. So follicle stimulating hormone can be produced by Graafian follicle. Then the organ is just the ovary. Then also this hormone is also produced by the pituitary gland. Then where in the female body is this gland situated? So we can just say where in the female body. So we are looking at the female reproductive system. So female reproductive system. System or organs, which is we can summarize by saying ovary. States any 2 other methods of contraception used in Zambia. So we can say condomizing, where someone uses a condom, so condomize. Then we can use contraceptive pills. We have also injections, we have safe days, stuff like that. Then question 5, figure 5.1.1 and figure 5.1.2 shows 2 examples of variation in humans. Okay, so we have this one where we have individual bars like W, X, Y, and Z. Then here we have a curve. And let's look at name the type of variation shown in figure 5.1.1 and 5.1.2 and give 1 example for each type of variation. So type of variation number 1, whenever you have these individual bars, this is a discontinuous variation. So discontinuous variation is where the difference is clear. For example, blood groups. Then also sex in terms of male or female. It's easy to see that this is a male, this is a female. Then apart from that, we can also look at the eye color, then tongue lowering, then type of variation for figure 5.1.2 is, this is now a continuous. So for continuous variation, it's hard to see the difference. So example, we have height, then we have body mass or body size. Then question B, what genetic factors control the type of variation shown in figure 5.1.1 and 5.1.2? So for 5.1.2, these are just the genes. So we have genes, they are responsible. Then for 2, we have environmental factors. Okay, we move on to C. Give 2 environmental factors that might influence the type of variation shown in figure 5.1. So the first one, we can look at diet. Like the body mass, the size of someone's body can change depending if the diet is bad. We know what happens. Then the other one is, if you look at climate change, also is the answer. Okay, we move on to question D, the last one. Using a genetic diagram, explain codominance in the inheritance of blood groups in human beings. So codominance in human being is just the blood group AB. So codominance is a situation where 2 genes try to express themselves at once. So A is dominant, so is B. So since these 2 alleles are all dominant, we have the situation of codominance. Now, since we want, our focus is, we want to produce AB, which is codominance. So let's look at symbols. So the symbols is just A and B. That's our focus. Then the parents phenotype. So to produce this kind of, we need at least in each parent there should be an allele for A and an allele for B. So my point is, for, let's say, for a male or female, AA can be used or AO can also be used. So let's just use AO. So here for AO, we are going to say heterozygous for A. B, let's also use heterozygous for B. So whenever we use the word heterozygous, it means we are going to have B and O, A and O. But if the case is homozygous, you use AA. AA is homozygous, BB is also homozygous. Then genotype, for the male parent, since we are using heterozygous, we just say AO. This is the genotype. Then for the other one, we are going to use BO. Now we can see that there is a chance of producing AB. So gametes, one will be A, O, B, O. Then offspring F1. So this one will give us AB. So that is our focus. Then this one will give us heterozygous for AO. Then here we have BO. Then here we have OO. So phenotype, the first one will say blood group AB. Here blood group A. Here blood group B. Here blood group O. So for blood group O to exist, we need to it needs to exist in homozygous situation. So this is what I wanted to share with you. So that's it for this video. Thanks for watching.